Production of genetically modified animals. The production of transgenic animals is a much more complex process than the production of transgenic plants, and not surprisingly, it is also highly controversial. To produce transgenic animals, a foreign gene is inserted into the genome of an animal oocyte, egg, that is then fertilized. The fertilized egg is implanted in a host female and allowed to develop. The resulting offspring are the transgenic form of the animal. The procedure has been used to produce transgenic fish, pigs, cows, rabbits, and sheep. Transgenic animals can be used to produce pharmaceutical products, such as human proteins, by using a recombinant plasmid vector. The vector contains the gene for the growth protein as well as the promoter that directs the expression of the genes in mammary cells. The recombinant DNA is injected into the oocyte. The oocyte is fertilized and implanted into a host female. The transgenic animals are the offspring produced from that implantation. The milk of female transgenic offspring contains human growth protein. Production of genetically modified animal. 1. Clone a human hormone gene into a plasmic vector next to a sheep beta-lactoglobulin promoter. This promoter is functional only in mammary cells, so the protein product is secreted into the milk. 2. Inject this recombinant plasmid into a sheep oocyte. The plasmid DNA will integrate into the chromosomal DNA, resulting in the addition of the hormone gene into a sheep's genome. 3. The oocyte is fertilized and implanted into a female sheep, which then gives birth to a transgenic sheep offspring. 4. Obtain milk from a female transgenic sheep. The milk contains a human hormone. 5. Purify the hormone from the milk. Benefits of GM animals are increased production of a particular product, higher milk yield in cows, greater muscle mass in animals used for meat, increased resistance to disease, manufacture of human proteins, such as antibodies, blood clotting factors, or important proteins for medicinal purposes, and producing of organs for transplantations, xenotransplantation.